Okay, let's get started. So we have about 3 to 4 lectures left. Hello? I'm going to slow down a little bit and give you time to digest what you already learned and also focus on the project. So I had some questions, received some questions regarding the fifth homework. Okay, so the fifth homework is optional, meaning that you don't have to do it at all. You know, especially for those people who feel you are doing well or even okay, you know, with the assignments so far. Even if you do the fifth one, you're not going to get a bonus because the total score will maxed at the maximum number, which is 100. So this is really meant for those students who feel they need to make up some of the missing earlier homeworks, some of the less than ideal performance. Then you can do it and earn extra credit. Okay. So my goal is to really, at the end of the day, hopefully give everybody an A in the class. That's basically the goal. All right. I'm not guaranteeing that. Okay. So you still need to do the whole thing. And yeah, I usually don't like to see outlier grades and stuff like that. So this is just an extra chance for you to make up some losing credits. All right. So we have about 3 to 4 lectures left. And today, I think I'm going to maybe stop presenting you we are going to deviate from the classical literature of graphic models and start to see some peculiar applications of that or some strange ways of using it, okay, or learning it. So today is one example. Instead of talking about graphical models, like a distribution or some loss function directly defined on graph, we're going to study another way of using the graphs. Okay, it's about how to use the graph to define constraints or structural prior knowledges into a different type of inference problem. And I'm going to use a very widely studied application problems as a driving example. And of course, by no means I want to say this is the only application such technique can be used. This is one of the straightforward applications, but there are many other fancy ones in computer vision, in natural language processing. So here is the problem. Okay, the problem is the following. So let's imagine we want to understand the genetic basis of the disease. Okay, it's a very fancy problem, and it is a little bit hard to formulate if you don't know some of the basis of genetic theory. So here is one slide overview of that. Imagine that I collect in some way DNAs from you guys, and you probably understand that the fact that we are all human beings, meaning that our DNAs are 99.9% identical. Okay, and much more different than those from chimps and other places. But on the other hand, the fact that we all look differently, okay, means that we, after all, are different at some small locations. And these are known as a polymorphism. For example, in here you can see this single site. Some individual had a T and some other individual had an A. And that might be a critical site to make a specific appearance or body metric show different values, such as hair color or stuff like that. Right? And these are called single nucleotide polymorphism or SNP. Okay, you can basically take this as a random variable, defined on a certain location on your genome, and then the sample basically can be collected over a population. And there are many other places you can find the same kind of variance. Then, out of, suppose that your population had a dichotomy like the following. Some are healthy and some are sick. And if, there is this perfect alignment that those people who all had a T in here are healthy and those who had an A here are sick, then you have a good reason to believe that these two kind of exact correspondence may be non-incidental. They may be causal. Okay, so this is known as a genetic association hypothesis testing. And in genetics, people call these causal SNPs. So this might be providing the genetic basis of a heritable disease, meaning that your disease is not due to you catch a flu or you eat something bad. It is because in your DNA you are intrinsically flawed. Everybody is flawed in this regard, but they can show different manifestations. Right? So the goal of genetic association mapping is to study this type of disease, which are genetically based. And you can imagine I collect, samples, populations, from healthy and sick ones. And then what I can do is the color isn't showing very nice, but you can see there are this light color telling you that I can do a testing one by one on each of these variations. By the way, don't get the wrong perception that the variations happen so frequently. Okay, every 2 sites you have a variation. This is impossible. In fact, every couple of tens of thousands of sites, you may have a difference, or even less frequent. If you have this frequency, then one of us must be a monkey, one of us must be a bear, or something like that. Right? So you're going to test all these sites one by one. I neglect those in the middle, which are identical. They are not worth testing. And then even for those variables, some are just giving you healthy differences, like eye color, hair color, skin color, and stuff like that. But maybe a few of them are truly, candidates of disease causal SNPs. And then you're going to scan the genome basically like this, use a scan statistic. And then maybe you can define a score for the scan for the correspondence between this and that. So what could be a score? You can imagine. The score can be arbitrary, can be mutual information maybe, right? Could be correlation. If this is not necessarily just a binary split, maybe some continuous variable, how sick you are, measured by a number, then you can even compute a correlation. So at the end of the day, you probably were able to claim that one of these is truly associated with the SNP or with the disease, and then you declare success. And a good body of statistical literature has been devoted to designing the right score, for this scanning statistic, and then even to compute the statistical significance of that. And we're not going to discuss that here because this is not a genetics class. And over those studies, no graphical model, no graph is involved. What we are interested about is a more complex scenario, in fact, a more realistic scenario of what we call complex diseases. So say a complex disease such as cancer, for example, is not just say, hey, you have cancer and he does not have cancer, stuff like that. Or maybe even not as simple as you have a lung cancer and he has no cancer or not. In fact, even if one is diagnosed with lung cancer, there are many, many subtypes, okay, which may even appear similar, physiologically, but they can be still genetically different. And also because of this, something called a spectrum, distribution of the symptom, it is very unlikely that there is a single causal SNP that is, getting people a cancer or not. Okay, usually it is a catastrophic failure in a system that is caused by many, many mutations or artifacts in the genome. Therefore, there are these multiple causal SNPs. Then the question comes, does the scan statistics I just described earlier still work here or not? One thing is that you can scan, one by one, and then now instead of picking the top one, you can pick the top 10 or top 20, and then call those SNPs. That's one way of doing that. So I want to ask you, is this intuitively a good idea or not? Maybe not, okay? So there are many reasons that maybe two causal SNPs are only going to show a significant statistical effect when they are tested together. And if they are not, if they are tested on its own, the marginal effect can be very small, right? So this is which we call the interactive features, which is non-additive, for example. And there are also risk of redundant features. Maybe these two SNPs, one is really causal, the other isn't really causal, but it's showing association. Why? Because these two SNPs are just physically too close to each other on the DNA. Therefore, once this SNP enters the descendants, that one also enters, because there is no room for these two to do random permutation just because of physical constraints. If you know a little bit of biology, this is a phenomenon called recombination. The recombination between these two can be rare, depending on the distances between these two. So what I'm trying to say is that a single scan statistic sometimes may pick up wrong signal and sometimes may miss important signals. Right? So a new paradigm of doing this complex disease genetic association analysis is to do multiple hypothesis testing, or maybe multiple SNP association analysis. And that's basically to take care of the complex combinations of effects on this side. And then on the other side, simply testing against healthy and cancer may be also too naive, because even cancer, of course, we know a few hundreds of types of cancer. Therefore, how come you can have only a binary indicator of cancer or non-cancer? When you are in the cancer, there are many subtypes. Therefore, such a discrete indicator may be insufficient. And also, it's not very helpful in terms of illustrating the mechanistic underpinnings of cancer. Right? So a modern way of describing the disease at least can have these two type of styles. One is to describe the molecular, the so-called molecular mechanisms of cancer, maybe scan the activity of your genes. Everybody of us may have about 20,000 genes active, and some are active because they need to react to a attack from the environment. Say I got infected by something, then suddenly a gene is bring up and react to that. So study this so-called gene expression network can help you to figure out what it is, what does a cancer mean mechanistically at the molecular level. Okay, maybe a particular module here, gets up, and that basically explains a particular subtype of cancer. Another module here is up, may account for a different type of cancer. So this is a much more interesting signal, to be used to investigate the causal SNPs. And even without the modern, technique for molecular biology, even when you go to a clinic, a qualified doctor wouldn't just ask you, do you have a cancer or not? Are you sick or not? They will actually ask you specific symptoms. They will measure your temperature, for example, right? They will maybe take a drop of your blood and measure a number of biometrics, right? And therefore, you have these clinical records of also high dimension. And that is often used to describe a whole disease. You have a list of features. So in this regard, a new modern paradigm of disease association is to find this multi-correspondence problem, where multiple candidate SNPs or genetic variations in here are jointly influencing multiple outcomes in here, such as a module in a network, or maybe a couple of symptoms in the clinical record, such as jointly your body weights, your blood pressure, and your other things, right? Your sugar level and stuff like that. So that basically argues for the new paradigm called the structured association, which is very different from this traditional approach where we do scan statistic and tease out single signals to a particular output. We're trying to now do this in association. Sometimes we call this is a genome, and that is a phenome, all the phenotypes. And then you're going to detect the so-called high bandwidth signal from the totality of here to the totality of here. Okay. So we ask what kind of technique you want to use. And again, here is just a graphical illustration. You have this thin line which point one dimension here, and do a pairwise statistical test, such as correlation, cross entropy. But in here, you can now do this high bandwidth, multiple signals here, causing something in multiple signals in here. Right. All right, so that is a problem. One thing that is interesting is that to make sure that the signal that you discovered is useful, for the people for the doctors or for yourself to take action on, you need to make the signal sparse. Okay, so this so-called actionable, information extraction is the holy grail of data mining. You can really dig out a lot of data at the end of the day. Unless you really want that data to go to, say, in Facebook, another classifier to spin out a signal. But if you want to consume the data by yourself, you really want them to be meaningful and sparse. This same thing also occurred in natural language processing or in image. You don't really want to get a collection of all signals. Say, okay, I dump you 10,000 topics, and each one have a weight, and they are all contributing to whatever content in your document in a certain linear combination. That kind of information is not digestible by human, right? You really want to pick out certain salient topics which are being used to generate a document. So genetics, work the same way. You want to get maybe a particular causal SNP in here, which are simultaneously causing maybe these few genes to act abnormally. And this is usually called a pleiotropic effect. And another scenario will be maybe multiple signals in here are jointly causing one gene to be functioning abnormally, and this is called epistatic effects. And you can imagine a combination of this. And these signals are really sparse signal, meaning that the red line or the blue line are out of a combination of a full bipartite graph where you have millions of signals in here and tens of thousands of signals in here. Everyone can have a line, connecting the two sides, but you only are interested in those truly significant ones. So sparsity is another need, in addition to just computing the association score. Okay. So what is the right paradigm for doing that? You can imagine. Okay. There are, of course, many different ways of doing that, but so far one of the mathematically most effective way of doing that is to do the so-called sparse regression. Okay. You all learned about that in your Machine Learning 701 or other places, which is to take a very modest kind of a starting point to assume that your disease signal is now called Y, which are basically here. And then your genetic causal elements are called X. And then you basically assume a simple causal model where a linear combination of the genetic effects is going to lead to a phenotypic outcome. Okay? And here the X can be encoded in different way. It could be a 0 or 1 binary signal, meaning that this particular genome site on the genome is mutated or normal, or something else. So we don't care about that genetic basis in here, at least. And you can imagine here are just two vectors, and you have this beta, which is the weight. And this weight, of course, is a very high-dimensional vector. Okay, J equals to the total number of candidate covariates or features in the whole genome. It could be as big as a few million, if not hundreds of millions. Okay, and then you do a regression, and we know that this does not give you a sparsity, and we want to have a sparse regression, meaning that we want most of the data to be pushed to 0, right? And we are hopefully very familiar with that technique already. We bring in a shrinkage function, okay, which is the L1 norm of the beta. And then this L1 norm can be actually translated, in its this is a constraint. It is an optimization problem. It's a regularized loss minimization problem, but the dual of that is a constraint optimization problem where you're penalizing this. That translates to constraining the beta to be bounded by some upper limit, right? And we know that this geometrically translates to a feasibility space of the beta in the form of a diamond if you want to take a L1 norm. And then the loss function, this part, the contour is in the shape of a ellipsoid, and the solution happens in the intersection of this feasible region and that feasible region. And then you can now visualize, or at least get the right intuition about why L1 leads to sparse solution, right? Because this feasibility region, even though it is still convex, it is having extreme point, such as these corners. And once a feasibility region has all the extreme point, then the intersection are more likely to happen on those points because they stick out, right? And therefore, if you get a solution in here, then that dimension was basically pushed to 0. That's becoming sparse. This is a two-dimensional solution, a two-dimensional illustration. If you are in very high-dimensional space, you can imagine this feasible region to be a strange animal with a lot of little corners, and then the one corner corresponds to a solution where many other dimensions are pushed to 0. Right? So that's basically a sparse solution that gives you certain advantage of directly detecting signal from here to here. And now the problem is that we may have multiple signals, right? For example, we want to have multiple outputs in here, and you can set each of them to be a regression like this, right? And then the coefficient is a beta vector. But if you want to solve multiple problems, you have multiple outputs, then you can basically make them as a vector output rather than a scalar output. And then if you still want to use the regression paradigm, then this is about a multitask regression problem, and the coefficients is the beta matrix, in which every these columns of beta correspond to one of the linear regression problem. It's one task, but you have multiple columns, and each row here corresponds to the strength of a particular genetic signal coming out of it. Okay, so this is the so-called the input-output regression, where you have a k-dimensional input and a k-dimensional output and a j-dimensional input, and your goal is to make this matrix sparse if you want to interpret the results. All right, so that's basically the setup of our mathematical framework to solve the genetics problem. And what I'm going to continue now is to give you a technique about how to use the graphical model or graph language to enable a smart execution of what I just talked about, how to detect those signals by leveraging on a lot of the structural knowledge or information you could get from biological data or from your domain knowledge. Okay, so here are the two main papers, and let's go visit each of them one by one. Okay, so here is the first problem. So I said that you have multiple outputs. For example, your symptoms or your genetic gene expressions can be multivariate. You have multiple symptoms, multiple gene expressions, and that basically forms a complex definition of a complex disease, right? Multidimensional definition of a complex disease. But the truth is that not all these different outputs. Okay, this is basically this gigantic Y signal. It's no longer a single scalar Y. It's becoming a vector of Y. The truth is that not all these signals are independent of each other, right? some signals are correlated, some are not. For example, here, and this correlation information can get from different places, can be obtained from different places. In here, this is a graph I personally obtained from a physician at UPMC. She is accomplished doctor, and she actually knew exactly how these different traits should be dependent on each other. For example, she identified that these 4 traits belongs to a subnetwork describing the quality of life of the patients. And this is a gigantic subgraph which describes actually this lung physiology. And within this lung physiology subgraph, you have 2 subtypes corresponding to different parts of lung physiology. Okay, now the interesting question is that knowing all this structure, this is the structure, it's a network basically over the traits, the clinical traits. And I want you to do this multitask association analysis. Do you ignore this structure or do you not? Or do you think they are going to be helpful or not? Okay, so that's basically the challenge. If you look at the statistical literature, up to very recently, people pretend that they don't even know this structure. Okay, even if the doctor know this, they will say, "Oh, don't tell me. I mean, you're going to confuse me, or I don't know how to use that anyway." So that will be left unused. And in fact, they will treat this whole thing no different from a collection of independent traits, and they're going to test the association one by one, and then give you the score. So there is a statistical flaw in that kind of practice, because if you are testing the significance of association to a complex traits with multiple variates, the hypothesis testing should be a multi-hypothesis testing. It is to test the association to multiple traits. The null hypothesis therefore there is not a null hypothesis built on every independent traits. So the existing technique isn't really building a null hypothesis based on this structural information. Right? So we ask, if we really believe what the doctor says is interesting, how are we going to make use of this knowledge? Or what's the implication of this knowledge in terms of detecting the signal from here and here? Okay, so that's the problem proposition. Any questions so far? All right, if not, let's find a way to see whether these graphs actually can be useful. Here I can share one information, okay? Suppose that one of the biggest challenge in this association analysis is that there are a lot of false positive signals, and there are also a lot of false negative signals. Meaning what? Meaning that maybe these 2 things, actually say suppose there is a signal in here. Okay. I'm supposed to draw something. How come it does not allow me to draw it? I'm not allowed to draw a graph on that. Very strange. Oh, okay. Right. So suppose I have such a signal in here. Not the right pen. Okay. And this signal somehow showed this particular genotype showed a signal associated with here. Okay. And somehow it also, you can test the strength of this association from here to this one and also to this one. Right? And these are 2 traits in here which is connected by an edge. So now I ask, if you run your lasso or run whatever technique and you find a signal in here, a beta and another beta, and one of them is very strong, the other of them is very weak or maybe nothing, how much do you believe this signal? That's actually an intuition I want you to derive yourself. Any insight? These 2 traits are strongly correlated. Maybe they are the same thing, by the way. So maybe this is body weights, and this is your height. They somehow, if you are the taller you are, the more body weights you will have, right? Roughly. But somehow a trait is associated with only this, but not that. What do you claim? Okay, so a total silent. Here I want to say a few side words. When you learn graphic models or machine learning type of thing, you don't just stay with the equations or math, and that's why I'm not a fan of deriving a lot of equations on the board, because you can do that always in the homework. The more difficult thing is actually to see how those translates to a insight, bring in real knowledge. For example, right now I was asking a very, very kind of a pragmatic insight. You use your graphic model already, the lasso technique or whatever fancy technique, and you find such a very interesting behavior, right? The signal is here but not there. What do you do? Right? Do you still trust your model or not? And if not, how do you modify the model? Right? That's how you actually learn machine learning. And here, let me share with you my insight. If these two signals are very, very correlated, but the causal signal comes only here, not there, that means that this signal is not trustworthy. It must be probably due to some random noise or some incidental kind of causation. Maybe because my instrument measured this trait when it was also measured in some other way. Basically, there are some other non-causal reason. Because otherwise, the same thing should also appear in here, right? I maybe just made a mistake in my experiment and detected wrong signal, right? So that means this information can be made useful, but now I'm basically offering you a way to post-process the signal and remove the signal. Now you can ask, can we make this information part of the model so that when I even estimate the beta, I don't suffer from such mistake, right? So that's actually a legitimate question. I can have another kind of interesting example. Say this particular signal. Okay, it is found to be weakly associated. Like, let me draw a different color. It is weakly associated with this signal. Let's say very weak. The signal may be 1/10 of the signal from here, or from even this round one. But it is also weakly associated with this one. Very, very weak. But they both appeared. Okay, and maybe even appear to a third one in here. Okay, if you run a threshold of sufficient test statistic or significance, they probably are way below this 0.005 threshold or some other magic number. So you are going to disqualify that. But somehow they co-occur, and they co-occur to three traits that are by themselves highly correlated. Again, what do you conclude? Well, three unlikely things to happen together is very, very unlikely, right? By chance. Therefore, they must have a reason. I probably should have a reason to believe this is actually a causal snip. For whatever reason, their signals are weak, but they are causal, right? So that's the kind of insight I want you to develop, when you deal with real-world problem, because it's never ideal like what you do on your UCI toy problems, right? And now my question is that how can we bring all this knowledge into the very design of your machine learning machinery, okay, to actually enhance your signal? Because post-processing is painful, and also it's also error-prone, because your original signal may already get lost even before you look at that, or just become too noisy so that you don't have energy to even scan all of that, right? All right, so that's the goal, and let's see how step by step, we are going to, somehow bring this kind of insight into a new design of the model. So again, let's revisit the basis. We have an association problem. We have the traits. We have the genotype. Imagine these are not character but numerical numbers, 1, 2, 3, and then you can write down a regression model from input to output, and your goal is to estimate beta. And if you just do linear regression, or even logistic regression, if you have a discrete traits, you will have an estimate like this. All the betas, okay, will have a non-zero magnitude. Okay, in genetics, they are called the effect size. And then you are going to probably write down a threshold to cut something away. But threshold is always an arbitrary thing, and also it is likely to include noisy signals, right? So again, as I mentioned, one way to enable feature selection in a consistent fashion is to use a lasso penalty, and then you will get this kind of estimate, okay, sparse one. And I'm not going to repeat the theoretical guarantees and the other advantages of this technique, which is established elsewhere. So my question now is to return to my earlier question. I have multiple traits. Okay, I have multiple traits, and also I knew that these traits or these outputs are interdependent in a certain way. Maybe these three are all belonging to allergy. There are just three different ways of measuring allergy, and there are two other traits, the FEV, FEF, which belong to lung physiology. By the way, these numbers are not made up. They are coming from the real data that we have. Okay, and then you can imagine the same way in here. Let's I can just mention a few words. In fact, all these genotypes or these input signals may not be independent either. Right? They may have coupling across some of them. For example, they co-locate on a particular chunk of the DNA, or so on and so forth. But let's just focus on this part first. And so if you have this known correspondence or association between multiple outputs, then instead of having this kind of signal, you probably will expect to see a pattern of this, right? If a signal exists in here, then there is a good chance that it should exist elsewhere as well, right? On the other hand, if it does not exist here, then chances for it to exist physically, this kind of pattern will be very rare. Some singleton block scattered. If you see such singleton block, you should be comfortable to remove it, unless they are extremely, extremely strong or whatever, okay? So the problem now is that how can we actually invent an estimator that gives me these kind of band patterns, okay? That is reflecting my prior knowledge of how different column dimensions are associated. Okay? It's a very, very straightforward question to pose. So how do we do that? So here I'm going to talk about our first graph-induced penalty function. In this case, it's no longer a graphic model. It's the graph-induced penalty function. We're going to introduce something called a graph-guided fusion penalty of the following form. So suppose that I have two outputs in here, which is known to be connected. Okay, this edge that connecting the two outputs can be from human empirical knowledge. I'm a doctor. I knew these two traits are measured or designed so that they are correlated. Or it can be a statistical estimation. Say these two gene expressions are correlated. I can compute a score to obtain an edge. But no matter what, if these two things are connected, I'm going to now introduce this penalty, which basically says that if the two betas associated on that, actually let me be a little bit more careful in here. There is a beta of j. There is a beta of j of m, j of l. M basically is this m, m's input, and j is the j's input, and m and l are this m's gene output and l's output. If these two things are connected, then I want to assume that their differences should be small. Okay, and they don't have to be identical, but if you want to be different, you need to pay a cost. That's basically a penalty, right? Just like here, if you want to be non-zero, you have to have a cost. And I put a sign here to indicate whether they are correlated or anti-correlated, right? So that's basically what this graph is trying to show. This is called the fusion penalty. You really, try to enforce the two things go co-zero or co-non-zero, but with the same magnitude. Okay, so the overall effects of this is what? So here I'm connecting these two traits, and I may have a trait in here. Okay, and what can we say about the beta in here? Okay, well, there is a path actually coming from here to here, but with lighter weight. So they can be still, actually transitive and maybe had a weaker constraint on the similarity of their weights. Okay, so in a sense, the information will be propagated across the entire graph, and by basically defining just pairwise constraints, according to the edge here, you actually achieve a global constraint on all the outputs, in your domain. Okay. So that's kind of the magic effect of this type of thing. And of course, you can reflect not only the topology of the constraint, but also the weights. Right? These edges may have a weight, a numerical weight of itself, telling you how strongly I believe they are correlated. And that actually just translates to this weight function on top of it, which is very similar to what I have here. So I call it graph-weighted field lasso. Okay? So at the end of the day, you can see this is the loss function. I have here the regressional fitness loss. I have the sparsity loss, and I have this is the fusion loss. Okay, I'm writing down now in a dual form of the lasso so that you can see them as the constraints, the box constraints. But you can also view them as the regularized regression problem, where each of its term is multiplied by a lambda, so they can become shrinkage function. Okay, so that's our first model, graph-guided fusion lasso. And the next is, of course, how to solve this constraint optimization problem, right? And that's something I would rather not to cover in depth in this class, because we have this whole class of convex optimization. This is a convex optimization problem, and there are packages, solving this using various different ways. I'm going to mention a few words about how to solve them effectively, because here our problem is a little bit different from those handled by the software packages out there. Usually when people talk about convex optimization in the NIPS paper or in the operations research paper, in my opinion, they are very small problems. They usually talk about a few hundred constraints and maybe a few 100,000 dimensions. And here, the problem can go as big as a few million. If you really want to go crazy, it can go to as big as a few billion, because I can make the problem arbitrarily complex by forming artificially complex features. For example, instead of looking each single genetic mutations one by one as the input, I can also have triplets or doubles of pairs of the genetic variations, and a million square gets you easily to a few billion, right? And basically you can make the input dimension very big. So that's a different problem we can discuss later, but I want to say that even though software packages are available, there is this interesting direction on how to solve such problem at extremely large scale. So here I have just a few kind of quick mention about one of the techniques we used. In fact, we used two techniques. This is the older one. There is a new one I'm going to mention as well. Is to just transform the problem using a variational technique. You probably will learn more about that in the optimization class, which I might teach in the later semester. So here you have, just to give you a flavor of how complex this problem can be, this is a regularized regression problem, and the regularizer here are L1 constraints. And this is a objective that is not smooth, meaning that they cannot be comfortably differentiated to form a gradient. They have this corner, okay? The shape of this loss is like they have a corner. And here, the gradient can have multiple directions. Some direction will crash you, okay? And therefore, people want to make it smooth. And one technique is to, maybe lift it into this problem, to square it, okay? And to square it is kind of okay, but it is still a little bit hard to actually solve, because you have this absolute sign here that is actually causing your problem to be still not differentiable. Okay. Then what you can do is to use a variational formulation to get rid of this absolute sign, to make them like this. But you need to bring in some auxiliary random variable in here and make it a optimization problem over some additional dimensions in here. And then, by solving this whole problem together, you actually can recover the original problem. So this is a complex story which is beyond the scope of this. But you can basically use this iterative procedure, fix that number, and then solve this, which is a totally differentiable smooth problem. And then you are going to re-estimate the auxiliary random variables and do iteration. So at the end of the day, you can get a solution. And here, so ignore these slides. This is basically say we are better. But I want to actually show you how to test the results. Yes?
A quick question. Why did we bring in djk into the equation?
Remember that in our graphical model lecture, we say sometimes we want to bring an auxiliary random variable to give you extra degree of freedom to escape certain difficult regime. Of course, in the spectrum graph model, we say that's not a good idea. Let's go spectrum and ignore all that. But that's for different needs. In here, you can imagine the following kind of geometric intuition. I'm in the corner. Okay, that is non-differentiable. I introduce one more degree of freedom and basically pushed on that direction. And things going over there, because of the magnitude of the dj is changing, I may smooth out a little bit. And things there can become differentiable. Okay, so this is a general trick. Which I hope maybe the other lecturers in the optimization class or myself sometime later in a different class can explain in detail. But I don't have time to actually talk about the specifics in here. Okay, so we leave that as a black box. So at the end of the day, I want you to actually pick another signal here in terms of how to test this kind of technique. Again, one of the bad practice in empirical domain is to cherry pick results. Okay, I applied a new technique, and then especially in biology, people like to do the following. Or maybe in topic models, literature, people like to do the same thing as well. So I got a whole bunch of results. I'm going to pick, I got maybe 1,000 results. I pick 7 of them or 8 of them, say, hey, they make sense. These genes were reported to be faulty, in the literature. Or maybe, hey, look at my top 7 topics. Aren't they beautiful or fancy or stuff like that? Right? So these are qualitative results, can be very attractive, maybe help you to publish a paper, but they really didn't prove the technique is correct statistically. Right? So you need to actually step back a little bit and test even a weaker condition that your method is actually at least doing the thing it is supposed to do by working on the data, you know the results. So here is such a full-blown test. The test began by a known hypothesis of beta. We actually know the beta already. Okay. Then we are going to also assume that we know the structure. Okay, in here. Okay, this is the structure of the outputs, the graph of the outputs described by the adjacency matrix. You can see there are 3 blocks in this output network. And then what you are going to do is you are going to start from the x, okay, and simulate it in some way. And you are going to use this beta and generate y, okay. And then, in fact, this beta are also produced from this graph so that you actually know how the beta should behave. And then you are going to remove the beta and you are going to, provide to our software the x and the y only, and plus this graph. Okay, so basically this guy plus x plus y will be entering our software. Okay, then you estimate beta to ask whether it is correct or not. Okay, and you have the ground truth of beta for you to compare with. And this is a very controlled experiment. You know everything, and if your program still is not able to detect that, okay, then your algorithm basically is faulty. And then in this regard, we are going to compare the performance of different algorithms, right? So as if you are dealing with the real scenario, you are going to now threshold the data in here to ignore maybe the weights, just the 0, 1 edge and none edge to form a constraint over the graph-guided diffuse lasso. And then you are going to test against a number of different methods. One is that let's just pick one x and pick one y to do this pairwise association test. You can do that, right? That's the worst possible, the simplest possible technique. And you can see, as expected, you get a lot of signals. Okay? And you can play by thresholding, and you can make a stringent threshold. You filter some signals out, you get this. And you compare whether this is quite consistent with this or not. Okay, the truth is that it is not. Okay, why? Because in here, your signal contains some noise, okay, and which actually can affect the recovery of the true signal. And then you run lasso. That's another comparator technique. Lasso means that I'm going to do shrinkage estimation from all the x to a single y, one by one. And but I'm going to ignore this graphical structure. I just assume that all the output y's are independent of each other, so I'm not going to use it. You get this? So the whole signal gets cleaned up a little bit, okay? And but it is still not quite the same as what you have in here, right? So for example, if I give you this, I will ask, is this a true signal? You probably will say yes, right? But in here you don't have it, right? And then maybe, what about here? I have a true signal here, but how come it's not here, right? So you have all these false positives and negatives. And then we run our method, okay? And you can see that, by comparison, this guy gets now significantly weakened, right? And you can basically remove it by a proper thresholding. And you have seen some bands which are consistent with the bands in here. And then you basically can compute a numerical assessment of the quality of the results. And that's basically what is done. You can now change the threshold of the method and then detect, compute this sensitivity specificity curve, and then you compare against the multiple other algorithms. And you can show that this graph-guided or graph-weighted one is giving you a better accuracy in a systematic fashion. Okay, so that's the first piece of message. Designing a graph-induced constraint and then turn it into a constraint optimization problem and then solve it, test it by simulation. And then here, of course, publish paper into run some real data. Then I'm going to ignore this part because this is quite biological and maybe less of an interest. It is basically applying this technique on some lung cancer disease data set. And we picked, in fact, the signal here could be very interesting, right? So you can imagine the permutational test based on traditional technique really gets you into a desperate regime. You don't get any signal, okay? Too many signals. Even for Lasso, it looks like quite sparse. You'll still have a few hundred signals in here. But in here, the graph-guided Lasso actually gave you some nice information. Especially, it is too bright here. There are two faint bands in here, okay, which actually should be discarded if you ignore their co-occurrence because they are very weak. But the fact that they co-occur as a band tells you that these are actually true signals. And in fact, we test these signals back to the physicians, and they actually confirm that these are signals that are of biological importance. Okay. All right, so with that, any questions before I move to the next story? Yes?
Mm-hmm. So say if you have two traits, they are highly correlated, and the true coefficient, maybe the beta 1 is twofold of beta 2, and in your fused Lasso penalty, you want the beta 1 equals to beta 2. So is it going to introduce some bias?
It is possible, but we can change the whole penalty function to reflect that kind of. Okay, so first of all, I almost want to dispute your scenario to be impossible, but even if that's happening, you can actually turn the fusion penalty to be a pairwise group penalty. You don't have to say the two traits are of equal weights. They can be grouped as a group Lasso penalty until it can generate the effect that they are co-zero or co-non-zero together. Okay, so there is a technique of doing that. So what I'm trying to say is that instead of having this penalty, you are going to use this penalty, which is it is the group Lasso, which is what is the group Lasso? I think it is something like that. Okay. And in here, you can roughly see that they will be encouraged to go down together as zero, or they can be all non-zero. But whether they are equally sized, doesn't matter. Okay, they will be penalized together. Basically, that's all we want today. But on the other hand, what you described is not very likely, because if the two traits are correlated, and if you properly normalize the data, by the way, need to be normalized, right? They are all in the range of 1 and 0. And it is very unlikely that you have a strength to here 1 to that 2. unless something miraculous happen. Right. Okay. So any other questions? There are a lot of arts actually in designing the penalty function, and which I'm not going to be able to cover entirely. I'm trying to deliver here a message that the graph helps you to strategize this kind of design and make them systematic. Okay, so what you see here is the next example. So just now I said the information is presented in terms of a graph over the covariates. What if this graph is not an arbitrary graph? It is a tree. That's another graph, but it is actually quite special because tree is usually used to organize the hierarchical clustering structure, right? And the branches are not just a pairwise dependency or pairwise distances, because there are latent variables in the tree as well. Therefore, the tree distances coming from here to here is of a different meaning, right? And then I want to say, what if I want to do a association mapping of signals from here to an entire cluster in the tree clustered outputs? Okay, and I give you the tree already. How can you enforce that kind of information? So what do I mean? It probably, actually you want to, that's another exercise you want to practice down the road. The doctor probably can say, if he can say as much as I just said, he's already a genius from a machine learning standpoint of view, or from a doctor's standpoint of view. Because usually they can only say, I know a cluster of the traits, and you do the rest. Then you need to translate what those actually mean statistically or machine learning-wise. So what do we mean by having the traits or the outputs grouped as a cluster? What do you expect to see or you want to see if such information is available? Only if you know that kind of knowledge, you can actually design the right model. And I didn't like a type of machine learning practice where I'm just waiting there to be told exactly, hey, design this particular loss function in so and so form. You do the math derivation at home. And that's not qualifying a machine learning scientist. It's really a graduate student's or maybe an undergraduate's algebraic homework, right? So here I want to ask you to design that particular function using the graph to reflect this kind of knowledge. So what would you begin with? Any suggestion? Well, this is not really very trivial, so I'm actually challenging you too much. So the author who actually came up with the idea is now a junior professor here. Almost because of these two papers she actually managed to publish. Okay. So here is an idea that I can talk about a little bit. So there are justifications about why tree is better than network. We can probably pass that. And we worry about how to use the tree to guide the lasso. Okay. So here is a kind of a high-level insight, right? So if you have a tree of traits or of output signals, say these two are under this tree, okay, by an edge of height h. Okay. By the way, the height of the tree usually describe the tightness of the cluster, right, under the branch. If they are very short, what do they mean? If the tree is very short, does it say anything about these two elements below the tree, or the leaves? They are not much different from each other. If I have a tall tree, that means they are very different, right? All right. So here is something I would like to actually suggest. I have two things which are now at least grouped under a tree. Then if they are under a tree, then I'm more likely to see this kind of pattern. They are showing up together if there's a signal. The signal here is each of these blocks correspond to a site or a genetic variations in the input. So this is X, right? And these are different Ys. Y1, Y2, actually G1, G2. Okay, and but if I have a tall tree, In fact, these three are very short, meaning that they are highly coupled. This is a tight cluster. These two things are very similar to each other. Therefore, they are more likely to behave the same. They are co-non-zero, co-zero. They can be occasionally, different from each other, but such incidents are not very often, right? But if these trees are very tall, almost suggesting they are uncorrelated or not, this cluster isn't really meaningful, then you see occasional co-black and co-white, but very often just individual white and black. So that's kind of the first translation. What does a tree mean in terms of their influence on the estimated beta? Okay. Then you want to say, okay, if I believe this is what it should be, then how can I form an estimator that encourages this kind of pattern from our estimator? Because when you do lasso regression, okay, on each of these individually, you don't have this constraint. Okay, they really can behave artificially. When they actually happen to be co-black and co-white, it's just by chance. Okay, you have not at least you don't have a bias or a preference on that. Usually when we talk about Bayesian statistic, we're always in the space of, say, without any knowledge, a priori I prefer that, right? So in frequentist domain, you have the same kind of needs. Without seeing data, a priori, how do I want the beta to look like? And this is basically almost like a prior knowledge. Without data, I want the beta to look like this. With data, I may be deviating, but I need to have strong evidence from data to be deviating. Okay? All right, so now how can we do this kind of inference? This is a nice idea. So suppose we have this tree, okay, and just of two genes. And that means these two genes are grouped under a cluster C. And then, the question is that whether I should select the two betas, okay, jointly or separately. Separately means that these two genes are not in the same cluster, and jointly meaning that they are in the same cluster. And jointly meaning that they should be either go to zero together or they should be non-zero together. Okay, and here is a way to enforce that kind of pressure. Okay, so on here I have the usual lasso penalty. Okay, the sparse penalty where I have a L1 norm on each of the beta. And what does this mean in terms of a selection on beta? What does this part mean? They are selecting the two beta separately, independently, right? Because there is each one can go to zero without influencing the other one, right? And then I have this, okay, and this is a L2 norm, which is defining, the kind of length of a vector. Okay, defined by beta 1 and beta 2 together. Therefore, if I want to push this down, I'm actually pushing both the beta 1 and beta 2 down, right? Therefore, they are jointly penalizing the two betas. And then, so I have now a joint selection and a separate selection. I'm going to now make a balance between these two forces. And the way I achieve that is to introduce the H. What is H? H is the height of the tree. And this is a very ingenious design. So your tree height is H, and if the tree is very tall, that means you really are assuming they are separate traits. They are not coupled together. Therefore, I'm going to encourage this part. Okay? If the tree is very short, okay, then I really meant these two traits or these two signals are jointly together. They are in the same cluster. They should be co-selected. Therefore, this part should take over. Therefore, I have 1 over H to do that. Okay. So this is the initial design of the tree-guided group lasso. So this is actually a group lasso, but now you can see the grouping is very, very interesting, right? You have different groups, and each group is defined by the tree of height 2. And if you look at the literature by some famous folks, such as Terry Tao and Candice and fellow people, they actually had a technique known before called elastic nets, which are roughly achieving this. Okay, elastic nets is exactly grouping versus non-grouping, and they have a weight. But this H does not have the meaning of the tree heights or the numerical value is actually unconstrained. You can do that. So we now have this h. In fact, h is a number between 0 and 1. It's a fractional height. But now the things become more interesting if I have a taller tree, like three nodes and 2 levels. How to do that? That's where things get very interesting. So I have a cluster of 3, okay, and then I'm going to recursively divide the cluster, okay, according to the tree until all reaches the leaf level. So I have here a subcluster of 2 nodes, here a subcluster of a singleton. And what I can do here is I have this one, okay, and this one. So this one is basically saying, okay, they are all under the same cluster. I'm going to now treat them together. But if they are separate, they should be separated into this group and that group. They have 2 groups. Okay, one group is a singleton, the other is something which to be defined on the subtree. Okay, and here you have a joint selection and a separate selection. And here you have a compound separate selection because this one itself is not a singleton yet. You're going to expand it because we know how to do the 2-node tree already, right? So this is basically what copied from the previous slides. And you can do this recursively all the way up to the entire tree. It looks very daunting because you have a lot of branches and others to visit. But the tree actually is a very economical structure over the network, over the nodes. If you have a node of n, this tree isn't too big. It's actually fewer than the number of edges in a complete network. Okay, so this tree summation here, even though you expand like this, looks hairy, but it is not as many as the network summation, which is over I = j, things like that. Okay. So that's pretty much one of the key idea of the tree-guided group lasso. You are going to group, recursive, not recursive, nestedly, different groups under the tree and weight them, according to the tree height. And this grouping, this weighting idea is also very clever. You may worry that maybe because of this recursive division and weighting, maybe some of the traits, some of the outputs get disproportionately weighted, and others may experience less weight. That's a possibility, right? Because every time you need to multiply h, something like that. Are they going to be fair, in terms of generating the same pressure? And that's actually a problem we actually experienced when we designed this model. And then there's a theorem proving that for every edge, every node, the total amount of weights it experienced can be actually numerically calculated, and they actually equal to 1. Therefore, for this beta, in every dimension, they actually receive the overall same amount of pressure, okay, cumulatively. And that is to contrast the earlier technique where there are this, maybe also tree-inspired grouping, but without the proper weight. Then you will see that some dimension received, overall less pressure than other pressure, than other direction. And then that creates an artifact in the selection procedure, just because your tree is putting the nodes into different branches. Okay. All right, so that's another lasso. Okay, so like this, the tree-guided group lasso. And you are going to, again, perform a convex optimization calculation to estimate the beta under this shrinkage. And we can use the same variational formulation as we had before. Okay, I'll turn to a black box software package to get that. Okay, so here the test can also be very interesting. And again, we start from the ground truth. This is the true beta. Okay, and you generate some data, and then you use other different algorithms to estimate. And of course, you can imagine the pairwise test is going to be extremely bad. So we started from lasso, which is known to be the best at that point. So you see this signal plus pepper and salt. Okay, all these other dark things. If you knew this is the truth, you know how to pick signal from here. But if you don't actually know the truth, then all these black dots are as qualified as the other black dots in here, right? So you cannot really tell apart. And this is the tree-guided group lasso. You can see the signal gets cleaned up at least significantly compared to here. All right, so any other questions on these two models? If I got time, I can talk a little bit about a computational acceleration ideas that can be also interesting. Questions on the model part? All right, so on the computational side, this type of optimization problems, even with the standard tricks people invented for variational reformulation of a non-smooth problem and block coordinate descent and stuff like that, you cannot go very far, okay, in terms of scalability. So here you can see, at about 200 SNPs, all algorithms, lasso or R algorithms are, getting a few hundred minutes. It's actually becoming very slow, okay? So why does things become slow? That's actually the question I want to ask. So the slowness actually comes from a number of different sources, right? So look at this convex function problem. It's very high dimensional, but furthermore, it's a constraint optimization problem that necessitates some not so efficient algorithm. First, this loss function is a non-smooth loss function. Therefore, you cannot easily take the gradient method. Right? Gradient is usually very quick, but here the gradient doesn't exist. And secondly, you cannot even apply a very nice gradient function because there is this weird property I don't have time to talk too much about. It is actually generating overlapping constraints. Okay, what do we mean by overlapping constraint? It means that I have variables, okay, and then I have groups. Here's a group, and then I have another group, okay, and these two groups are overlapping. And when you do a coordinate descent, and you come to here, you actually ask the whole thing to go one direction based on a group constraint in here. Then you actually will be revisiting this same coordinate under another group, which will tell you to go the other direction. Okay, there are these kind of intrinsic inconsistency possibly generated by an overlapping group class. Therefore, some kind of a joint optimization problem, you need to bring these two groups together when you are trying to optimize that direction. And that actually creates a not so efficient algorithm. So these are well-known kind of limitations of convex optimization problems. And here I just want to give you some quick ideas about how things can become a little bit more manageable. First is you want to introduce some kind of transformation so that your overlapping group constraints can be variationally defined as a linear transformation, okay, so that these overlapping groups are somehow deconvolved into non-overlapping ones. Okay, there are a number of ideas. ADMM is one example where you just introduce dummy variables to replicate constraints to make them separate, and then put constraints on the separated part to make them identical. But here you have a rotation. Somehow, if for example, if you see a knob from this direction, they may be a knob. But if you see the other direction, they may be actually not a knob by another space. So you do that and basically make them non-overlapping. But non-overlapping constraints can still be non-differentiable because they have corners and all that kind of thing. And what we can do is to introduce some auxiliary function to this is called a proximal kind of operator to generate an approximation as a function of some auxiliary functions. And then that makes the whole loss function differentiable. One can prove that this new loss function, okay, can be actually differentiated, and then you have nice properties to ensure that this gradient is convergent. So here again is the high-level picture. You have this original loss function, and which has this very kind of annoying kind of singularity point that is non-differentiable. But if you introduce some kind of adapter functions, okay, in here, and suddenly you actually make that point smoother, okay, because it adds one more dimension into. So here is the alpha dimension that you basically make this loss function stretched out in one dimension. And then when you look at the whole loss function, it becomes very smooth, even in that point. Again, these are, technical details. As a result, you actually make this whole loss function non-overlapping and differentiable, and then you can prove that a proximal gradient method, under that kind of a transformation, leads you to very fast converging algorithms. It's a first-order algorithm with a convergence rate of 1 over epsilon instead of a 1 over epsilon square algorithm. Usually use the interior point methods that is popular for optimization. And it has the nice complexity, dependence on the input dimension and output dimension. Okay, so now I want to wrap up by some empirical plots, okay, on designed experiments so that you can appreciate the speed up. So it is now compared on two types of problems. One is the GF lasso, and I believe the other is the tree lasso. And three candidates. One is a quadratic programming algorithm. And you can see we want to measure the complexity or the cost over bigness in 3 dimensions. One is K, the number of outputs, anywhere from 0 to 10,000. Now it's getting really big. You can handle 10,000. And here it is the input dimension from 1 to 10,000, 0 to 10,000. Here n is the number of samples. And you can imagine, the quadratic programming technique that goes like that. Okay, basically, if you go just a few dozen or maybe 100, the cost is already prohibitive, right? But you can see this very smooth kind of increase of the time complexity. Even at 10,000, it is still manageable. Same thing here. The SOSSP is a second-order co-programming. It is a slightly advanced method than the quadratic programming, but it's still second-order. Okay. And on the input dimension, again, it goes quite nicely with the dimensionality of the problem. And on the input data size, this is a logarithmic plot, and you can see the cost. Okay. And the same thing on the tree lasso, and comparing SOSP and the proximal gradient, you can see this is a very different dependency of the complexity. All right, with that, I'm going to close by saying this is not quite graphic model, but you can see the graph is a nice tool to inject information, not only to probabilitists or Bayesian statistician, but also to a pure frequentist framework where you don't even use graphic model, but use some loss minimization procedure. And here the graph can be used to design clever loss functions, okay, to introduce constraints and other biases, against your estimator to achieve the desired results. And this domain is rather popular in machine learning these days, mostly in the theoretical space, because shrinkage estimator and sparse estimators have very nice statistical property of being convergent, and the people can prove nice asymptotic results on that. But on the other hand, the scalability and the utility of this method has been not widely practiced because of the lack of such structural constraints to allow people to design flexible models. So what I'm showing here is one way of hopefully making such model more flexible as other graphical models people would use in many applied domains. Okay, so any questions before we dismiss the class? If not, we'll see you next week.